Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello and welcome to this episode of the Epigenetics Podcast. Today I'm happy to welcome Christa Buecker from the Max Perutz Laboratories in Vienna on this show. Please let me briefly introduce you to our audience. You got your PhD from the University of Utrecht in 2010. You then moved on to do a postdoc with Joanna Weissocker at Stanford School of Medicine, and you joined the Max Perutz Labs in Vienna as a group leader in 2015. A question I like to ask every guest to start off our little podcast is, how did you become interested in biology in the first place, and then in pursuing a career in science?
So I think there are two possible answers for that. On the one hand, I grew up on a farm, and I think then you're fairly early on exposed to kind of genetics and how the environment shapes what you're doing. And I think that was because my mom was a biology teacher. That was always something she would then also explain to us kids. And then I had some really amazing teachers in my high school, one in chemistry and one in physics and math. And they really encouraged me to go for a study science. And they also drove me towards biology, making sure that I understood that this kind of like brings in all the different natural sciences together. And so their influence was really strong in my choices for studying and then becoming a scientist or starting to study biology.
So it really seems that teachers have a very big influence on science that is done nowadays.
I think so too. I think that we all profit so much from them and get so much from them out there. I see it with my son. I see it with a lot of kids right now in school, how much the teachers are influencing them. And I see that with myself. So I think they do have a huge influence.
So let's talk about your science that centers around transcriptional regulation during early embryonic development. I want to start in the year 2010. There you demonstrated that human iPSC reprogramming in the presence of LIF yields human stem cells that display morphological, molecular, and functional properties of murine ESCs, so embryonic stem cells. Could you talk about this study, how you got into this, and what you found there?
Yeah, so that was my PhD project. And we had been long interested in what is mouse pluripotency, and we worked mostly with murine embryonic stem cells, mostly to study germ cell differentiation. And that was the original start of my PhD. But then during my PhD, which centered mostly around stem cell biology, reprogramming was discovered by Yamanaka, and there was this huge influx and flurry of papers happening. And so we then started to, of course, also play around with it a little bit, and took some human cells that were reprogrammed, but they were really weird, and some clones really displayed some very interesting morphologies. And we compared them, because we had much more experience with mouse embryonic stem cells, that they actually looked like mouse embryonic stem cells. We cultured them under similar conditions, and we could then show that they shared some things and had some things in common with mouse embryonic stem cells, but not completely. And so they were more in the direction of this elusive, or by that time, elusive, naive pluripotent stem cell state. And not completely, there were many things missing. But we could, the good thing about these cells were at the time that we could do homologous recombination on them, which was a big step. Now looking back with CRISPR and Cas9 being making this fairly easy. By that time, generating any kind of modified human embryonic stem cells was not possible at all. And so this was actually then the main driver for this study. And we could show that we could modify human embryo, like the human pluripotent or human cells, and that we then could also change the culture conditions again and turn them into cells that behaved like human prime pluripotent stem cells. And so this difference between naive and prime then became a much bigger interest for myself, also what makes these cells what they are. But it was a study that really shaped some of these ideas of naive pluripotency. And then, of course, there were many other labs that followed with defined transcription factors and defined growth factor cocktails that really then brought these cells into what they are today and that people use.
Yeah, maybe we can push the differences between naive and prime pluripotency for a few minutes, because you just said that there is like a difference between human and mouse pluripotency. Did I understand that correctly? And why is that?
Well, so there are some species-specific differences. And for the mouse, there are much better to take on a transcriptional state that is much closer to what we see in the epiblast of the blastocyst. So this around E4.5, that's where really the mouse embryonic stem cells that were originally derived. And people were not able, under these exact same conditions, to derive human pluripotent stem cells. All of the cells that they had generated from blastocysts, they were just differentiating away, and there was nothing really left there. So it was assumed for a while that maybe that is not possible. But then some labs have shown that different culture conditions can lead to human embryonic stem cells. So, and by that time, it was assumed this is really just a species-specific difference. As mouse is mouse, human is human, and so they are just not very similar. But the study from Paul Tizar's lab and Ludovic Vaillet, both of them showed that, or two different studies showed that you can actually derive mouse cells that behave like human cells. So then you had like this difference between them was actually not a species-specific difference, but it was a difference in the culture conditions. And that humans, and then that's where this race started to make human cells that behave like mouse, because they were considered to be slightly earlier in development, and thereby maybe also more prone to do more different, to turn into more cell types and have a higher potential for going germline, for example, or also making germ cells, which prime pluripotent, at least in mouse, don't do so well. And so, yeah, there is this difference, but it's not so much, there is some part of it is species specific, specifically for mouse. The mouse embryonic stem cells, the naive ones, they're just awesome to work with. And I still love working with them because they're so easy to culture. Whereas human is now much better. When I first started working with them, it was awful because the culture conditions were not very defined. They were differentiating all the time. And I had to clean up the cultures constantly to get rid of differentiated cells. And it was a pain.
So does that mean that there are species-specific Yamanaka factors, or are those universal?
No, the Yamanaka factors are pretty universal as far as I know. I haven't worked on reprogramming in a very long time, but I think you can take the 4 factors in a lot of different species and generate pluripotent stem cells. But it depends on which growth factors you're using then in combination. So it's not just the Yamanaka factors alone. I think we often forget that you just don't throw on these transcription factors and they do a job. And then you get something. But there's also the surrounding external culture conditions that are really needed for keeping these cells and generating a certain state that you actually want to work with, or that you can work with. So I think that is not to underestimate.
So let's circle back to the question that I just pushed back. And I guess this was during your postdoc. You showed that transition between the 2 pluripotent states, naive and prime pluripotency, is associated with widespread OCT4 relocalization, mirrored by also global rearrangement of enhancer chromatin landscapes. And this is right in our ballpark, right? Because this is epigenetics. So could you talk about those 2 pluripotent states and what you found out about these enhancers?
Let me throw in a third pluripotent state, because the original study we called it prime, but I think Austin Smith would rather refer to them as formative. So they're kind of like in between because they're transient. But in the end, I think we're like, it's a little bit also of semantics, what you call these states and what they are. So when I joined my postdoc lab, I wanted to look into what is happening during differentiation and what actually defines states. So the transcriptional response to changes in growth factors was pretty clear for me to use as a system. And I spent about half a year to figure out a system that we could use and that would be really ideal to study the changes in transcription. Because it's actually not that easy to find a system where you go from one state and then differentiate the cells into a slightly into a different state where they can't go back, but that is close enough that that you can really follow the cells over time. For many differentiations, you actually go, if you take a mouse embryonic stem cells or any kind of pluripotent cell and you differentiate them somewhere along the neural lineages, you have multiple intermediate states that you have to cross through. And so I was playing around a lot with different differentiation systems until the Hayashi paper came out, which I forgot now the last author, but they had this beautiful differentiation system into germ cells. But their first step, the very first step of differentiation, was taking mouse embryonic stem cells and differentiating what they called them into epiblast-like cells. And so this took 48 hours and the cells were not able to go backwards. And so we took that system and we're like, let's see what are the transcriptional changes during this and how are they regulated? And so we started to map out the enhancer landscape, which by that time also, this is years ago, was something not so many people had done. Because we
what kind of assay did you do?
So we did ChIP-seq at the time. We used P300. ATAC-seq wasn't developed yet. And DNA's hypersensitivity mapping was hard and very few.
Compared to ATAC-seq, you had to titrate everything and do all of this, all the timings and clean up, do all the gels.
Yes, no, it was the way that we did it was combining ChIP-seq for P300 with OCT4, which we knew was one of the main transcription factors involved in this early embryonic, at least in the starting population. And then, of course, K27 acetyl and K4 monomethylation as the two flanking ones. So what by that time we considered was an active enhancer. K27 acetyl, K4 mono, and P300 as the center. So we focused our peaks around that. And we had OCT4 included originally because we had the antibodies and they were really good by that time. We saw fairly fast that OCT4 can be found in both states, and that it also changed its localization pattern, which now in retrospect, of course, makes a lot of sense because it's a transcription factor. It works with other factors together. But because of the Yamanaka factors, being part of the Yamanaka factors, it was considered as one of the main pioneer transcription factors that would come in and then reorganize, or that would then go to one specific site and sit there. But we showed that is not quite true, that it does change its organization and its localization within the genome, and that it does work together with the existing transcription factor landscape, which I think is a nice addition to our understanding how transcription factors work in different cell types, because we're reusing the same transcription factors over and over again in many different contexts. And so OCT4, even though it is somewhat special, still does the same thing.
So does it replace other transcription factors at sites, or does it act in symbiosis with them?
It acts in combination with them. So in the undifferentiated state, so in the naive pluripotent stem cell state, it works with SOX2, NANOC, ESRB, all of these additional factors And these additional factors, many of them are not necessary for pluripotency. So they are somewhat redundant with each other, but OCT4 at the center is needed for the pluripotent state. So without OCT4, nothing is happening. But then it works with these accessory factors in one state. And then during differentiation, these accessory factors, many of them are downregulated. OCT4 itself is staying at almost the identical level. And then it goes to new sites together with new transcription factors. So one of these factors is OTX2, which was very surprising to us because it's actually involved in the retina and the neuronal differentiation. But it was one of the main factors that was upregulated and that had a motif very close to OCT4 binding sites. And if we overexpressed OTX2, it was able to grab OCT4 and bring it to new sites. So it really was one of the factors that drove OCT4 to new sites. There are definitely others. We still haven't, there are few of them identified. ZIG3 might be one of them. But we're still not completely understanding which are the other factors that might be involved there.
Did you also look at transcription and how this correlates?
It correlated very well. So whenever we had OCT4 going to new sites, the surrounding genes were activated. And when sites where OCT4 left, these sites were downregulated. Even when we overexpressed OTX2 and brought it in, we had a very strong correlation between the sites where OTX2 was now bringing in OCT4 and the sites where OCT4 left, because that is also so. The transcriptional changes were very, very clearly driven by this reorganization
and immediate.
Yes, fairly immediate. Fairly meaning that it's not a Drosophila where things are happening within hours, but within 24 hours, we saw of just changes in the growth factor environment. The cells cross over a point of no return, and then they can't go back.
Then you dissected the functions and interdependencies of 5 enhancer elements that together activate FGF5 expression during exit from naive murine pluripotency to understand how individual enhancers collaborate to activate transcription. Mm-hmm. Was this done your own lab already, and what did you do there?
So that was my the first study that we published from my own lab. And it was actually an extension originally from what I had done in my my postdoc work. Because we had identified the FGF5 cluster as one enhancer cluster that got strongly reorganized. So there, I think already in the original cell stem cell publication, we had FGF5 cluster as an example for the changes that we see on the chromatin.
When you say cluster, what does this mean in terms of members and like size in megabases or kilobases? I don't know.
Oh, this is kilobases. So okay. This is all within 100 kilobases. So it's not massively big. It's not the Sonic Hedgehog enhancer or the SOX9 enhancer where you see megabases of distance between the promoter and the enhancer. But still, there are 5 elements that are individually activated. So they are closed off before differentiation, at least in the ATAC-seq, we didn't see anything. We also looked at DNase hypersensitivity at these sites. There is not anything open there. But then during differentiation, OCT4, OTX2 come in, they bring P300, K27 acetyl, K4 monomethylation, and the whole cluster and the whole group of enhancers are activated. And we wanted to know how are the individual elements contributing. So similar to what Doug Hicks, for example, had done for the isoglobin locus or other enhancer elements, we now had a system in our hands where we could differentiate fast, right? We didn't have to go towards the erythrocyte lineage, but we could just go 48 hours and follow over time how are these individual enhancers activated. And so that's what the first PhD student in the lab started to do. He deleted individual elements and then combinations of elements, and started to look what is happening over time. How are these individual elements contributing? And we showed that there are some elements that contribute more and other elements that don't seem to do anything. But I think the most surprising part from that study was that none of these individual elements had any kind of function or any kind of intrinsic activity, which I think if we would have started from that point, looking at the intrinsic activity, we might not have dissected that locus. Because what we showed was that...
What does activity mean in this case?
Activity means driving a reporter promoter, like driving a luciferase gene with a minimal promoter. We also tested it with the endogenous promoter, so it didn't have much to do with the promoter choice. It was just that individually, those elements were not able to do anything, which by the time was also for reviewers was like one of the biggest issues. And for me as well, when I first did that, I was like, okay, so we don't have any activity here, but we still see when we deleted them, we saw changes in expression.
So when you delete all of them,
changes.
There was a change.
There was a big change. So we individually deleted each of the elements, and some of them we saw a 10 to 100-fold reduction in expression. But the element themselves didn't do anything. Mm-hmm. And so that was surprising. But I think it's also really, really interesting because it means that what we're measuring in MPRAs might not necessarily reflect what is going on within the genome, and we're actually having a context of multiple elements together. The second really nice finding from that study was that so when we tested these individual elements at the endogenous locus, we found that they often work together in a really super additive fashion. So it's not that you have individual contribution from one enhancer and from these other elements, and then you just put them together, and then you get, the additive value as expression. But in fact, what we saw is that they're often, like with all the things that we tested here, they often work in a really highly super additive fashion. So the output of expression afterwards was much, much more than the individual ones. And so that I think was also really interesting finding, because it helps us to think about maybe the cooperative part of these enhancers.
Are those enhancers located in one TAT?
In this case, they were located in one TAT, and they were, they're localized within one domain. Yes.
Mm-hmm. So next you showed that the interferon regulatory factor 1, so IRF1, is directly controlled by the murine formative pluripotency gene regulatory network and transiently upregulated during the transition from naive to formative pluripotency. You're just smiling. So maybe this is a...
That study actually goes back because we wanted to study something totally different. But I think that's how sometimes you design your reporters and your experiments in a way that you then end up with something completely different. I said earlier that we don't really know how OTX2 localizes OCT4, right? And so this study actually started based on that question, because we wanted to know, are there because one fact that I didn't tell you was that OTX2 can be knocked out as well. And it's not needed for the exit from naive pluripotency. So if you differentiate cells and then an OTX2 knockout background, they're fine. So there must be other factors, and we wanted to find these other factors originally. And so we designed a strategy to screen for factors that might be redundant to OTX2, sorry. And Merit, a student in the lab, she performed that screen, and we didn't find anything. So it was very, very disappointing. But we then looked into so anything that was different between the knockout and the wild type cell. So that was actually the start of the screen. But then we looked into the wild type cells, and we found that there was one factor that stuck out to us, and this was also always think about how to design your reporters. We had designed our reporters based on enhancers, but these ones that are active and that do have an effect. And the OCT6 enhancer was one of the ones that we used for this as a readout for the cell state. So the OCT6 enhancer is beautifully upregulated during differentiation, and we coupled it to a fluorescent reporter so that we could sort out cells using FACS that might not have upregulated it as much as the wild type and as the in this CRISPR screen. And so we performed the CRISPR screen. Then we had like one big hit that completely abrogated what we thought was differentiation. And that was IRF1. This was something that we had first put to the side because we didn't want to deal with any innate immunity. Also because mouse embryonic stem cells are not really having a good innate immune system. There is no innate immune response at this time point. So we were like a little bit puzzled by the result that we found this transcription factor. But but then when the other screen, or this combinatorial screen of wild type versus OTX2 knockout didn't really yield anything, we went back to the first result, and then we focused on IRF1, and we did the knockout and we characterized its behavior, and it turned out that IRF1 is actually a really interesting factor because it by itself, so it didn't regulate pluripotency, which was like my original hope as a stem cell biologist. We would find something here interesting. So that's not what we found. But what we found was that IRF1 itself was upregulated during this transition or during this differentiation, and that this transition itself, or this upregulation, was driven by OCT4 and OTX2. So these factors that are reorganized, they're binding to an enhancer close to IRF1. IRF1 normally is stimulated or is expressed through interferon, through the interferon response. But we didn't have an interferon response. So this was a developmental upregulation of IRF1. And we speculated that this might have something to do with protection against viruses. Because IRF1 is normally one of the factors that is then sensing and is upregulated during viral infection, and then protects other cells through a downstream cascade of factors from viral infection. So we tested that, and it turns out that the knockout cells, the IRF1 knockout cells, they were much, much more prone to viral infection than the wild type ones at this early stage. And so I think that was a serendipitous finding in some ways, because we would have never gone there without this screen.
Mm-hmm.
And we also found out why we actually got it, because the cell state was not changed, but IRF1 itself had a very strong binding site in the OCT6 enhancer. So our reporter was actually not reporting on the cell state, but our reporter reported on IRF1 presence or absence. And so that's how we got there. But I think we had a really cool story in the end. Like, long story short, I was very proud of that paper because it took many turns and many different parts in there. But I think it's a beautiful little story about how one finding can get you somewhere completely different.
Interesting.
And I would have not thought that I would end up with viral biology and innate immunity.
So imagine you're due to submitting a grant proposal tomorrow. What would you have written into that?
Oh, that's very simple. That goes back to this FGF5 story, because in the meantime, we have started to build a system. So the FGF5 story was a story where we took a top-down approach, right? We identified an enhancer cluster. We then deleted each one of them, and then we wanted to figure out what it does. And And we still don't know what are the transcription factors involved here. We still don't know how these elements are really working with each other. And so after we published that, we took some time and we built now a locus that we can use for integrating individual enhancer elements at different distances to a promoter. And we have a preprint out if you want to take a look at that, but I'm very happy to promote that.
I didn't see that. I'm sorry.
That's okay. So there so now we have a system where we can take a bottom-up approach. So we can integrate individual enhancer elements alone and in combination, and interrogate how they affect gene expression. And I'm right now we're going crazy with different experiments that we can do. The first experiments that we've done now are showing that we can integrate that. The distance of an enhancer to a promoter, of course, matters, but it also depends on the intrinsic strength of the enhancer. So the stronger an enhancer is, the further it can go and activate. Then the second part of it is that we can combine multiple enhancers together into our locus and then interrogate and then show that most of them, not all, but most of them work in a highly super additive fashion. So you place in one, it does do a little bit. You place in a second, it does do a little bit. But together, they can really support each other. And oftentimes, it's really these weak enhancers that come in and that help and support. And so my grant proposal right now that I actually submitted would be that we would really make this into an MPRA, but for distance dependency. So test whole libraries of enhancers at different distances, and then test out what is the underlying rules? What are the transcription factors? Are there some that do work with each other, some that don't working, some that determine distance dependency? So that would be the part that I would be writing right now if I would write it again.
So what defines the strength of an enhancer? Is it like the motif, how many proteins or molecules are bound to it? What does it define?
So my very simplified model would be that it's really the amount and the difference, the amount as a first part of transcription factors, but then also how many different motifs you have. And how many different motifs you have. I cannot put that into a quantitative term right now because we don't have the data for it yet. But I do think that you really need to have, if you have just the same one, you probably don't get a strong expression. But really a selection of different transcription factor motifs might be one. And then, of course, that leads to the recruitment of coactivators. And those then really are doing the job, right? The transcription factor itself is just a recruitment factor or a bridging factor. And so, that would be my main answer, but I will hopefully be able to tell you more in the future about what it is, because right now we can only say that there are many, many weaker enhancers but they can do something. They have a job, right? It's not that they're totally unimportant. But I think that specifically for short range or for bridging interactions within the genome, they're probably needed.
Okay. So for the last 35 minutes, we have been on a journey through your scientific career. Did we miss something important, or would you like to add something?
No, I think we covered, now that I threw in this last study, I think we covered most of the parts. So, yes.
To finish, what is your definition of epigenetics?
Oh, God.
I mean, this is now a new question I just added.
Okay, so when I actually have a slide in one of my lectures on this, because I try to make students aware that if they talk about epigenetics, that they should ask they should be aware what other people actually understand by it. So when I talk about epigenetics, I probably mean everything that is post-translationally modified on chromatin. I think that would be, or most things, that would be probably the closest to it. Because I would include like the acetylation of histones under epigenetics. Many people would not, because it doesn't get transmitted through the through mitosis, right? So I think it's a very, very hard, it's a hard question. Thank you for throwing that in there.
About the microRNAs count?
Well, not in my definition, but now that you throw it in, it probably is some kind of, probably, but not under my definition.
Okay.
But I just gave you my definition.
Exactly.
And, like it's, I tell you, it's so hard.
Yeah, exactly. So thank you, Christa, for your time and for being on the show.
Thank you for having me. That was fun.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening, and stay tuned.